Ovary cancer, less than 10% of patients are diagnosed at stage 1. We're very interested in looking at normal to tumor transition. So now we could actually determine at what point in that normal to tumor transition does this microRNA that we're interested in get turned on. Like, is it in the precursor cells, or is it later? Then that would be a way to detect these women at a very, very early stage. If we show certain microRNAs are highly expressed in patients that tend to do really poorly, and that these microRNAs are targeting signaling pathways that we know are detrimental to the cell, can we target these microRNAs? That's where we need to know the basic biology of how these microRNAs are processed.
Our laboratory's mission is really to understand what drives lethal human prostate cancer. And we specifically focus on transcriptional regulation and epigenetics to understand more about how RNAs are turned on and off and how that process impacts resistance to therapy and also tumor progression. And the model we focus on specifically is advanced prostate cancer. A physician scientist, I'm a medical doctor. I still see patients a half day a week. I have the privilege of doing that, and it really informs every facet of what we study in the laboratory. So we can ultimately get to therapies that we move back to clinical trials so we can improve the quality and quantity of life for patients. That's really the driving force of what we do. And I think without the inspiration from patients, I think we would not be able to focus on the right sorts of questions. And so I really view patients as our allies in all the research that we do, whether or not we're using patient material or not.
It was a fortunate situation where I had a mentor who was allowing me to be somewhat independent and explore my passion of pursuing a field that I knew I was personally affected by and wanted to continue working on, looking at the genetics of ovarian cancer.
Just the welcomeness of people and the passion people have for excellence at U of M. And I think that really comes down to the fact that so many of us are interested in trying to move the needle on the fields that we're studying with the ultimate goal of trying to help mankind.
One of the major reasons I came to U of M was to work with talented RNA biologists that can help catapult my research to the next level and utilize the clinical samples that we've been able to collect over the years, and also the mouse models and patient relevant mouse models that we've been able to generate to see if we can interrogate what are the most relevant microRNAs in ovarian cancer biology.
That I interviewed at U of M, I was really struck by not only the excitement of people's research programs, but how down to earth people were, their willingness to collaborate, and the collegiality. And I've been at, I believe, 5 medical institutions now and scientific institutions as part of my training. And I've never been at a place like U of M, which is really such an open place, even though it's such a large research university, and where the overlap between people who are kind, bright, and capable is completely overlapping.
We have a very strong group here who already is very collaborative and works with many people across the globe. So we're working together with so many different groups to see if we can build that cohort.
I think it's really the opportunity to interact with people for whom you may not have a preconceived notion that they would be a great collaborator or intersect with your work. That's really where I found some of the best discoveries and the best collaborations that I've had have been made. Blown away even to determine that there was a center for RNA biomedicine. And I think there are very few universities that have such a center. I think what is exciting in terms of understanding and learning more about the center is just the breadth and depth of science that's happening, and hopefully find ways to collaborate and tackle some very important problems in advanced prostate cancer, and also to help other people with the work they're doing, because our lines of study may have relevance to other diseases as well, no doubt.
Is the number of resources that are made available to scientists that are interested in exploring RNA biology. And that's, I've only been here 2 years and I've already been able to utilize this, such as the BRUCE core. So we collect a lot of patient samples. We also generate unique models of drug resistant and drug sensitive tumors, and we're able to assess what are the nascent RNAs that are relevant in these models using BRUCE. Training was really in tumor suppressor genes and oncogenes. It wasn't on microRNA, but fortunately, I worked with a lot of great collaborators in the microRNA field and was able to continue on and continue to this day working on microRNAs.
We're always open for new collaborations. Certainly those who are interested in cancer research and prostate cancer research are those that might be most closely aligned with what we work on. But many of the mechanisms of transcriptional regulation, splicing, and epigenetic regulation are certainly broadly applicable to other diseases. And so I look forward to learning more about the research that's happening in the center and meeting more people from the U of M community and finding ways that we can work together. Some of the best collaborations I've had over the years have been people in very disparate fields that didn't necessarily have a cancer background, but had approaches or techniques or methodologies that were very applicable to the research questions that we were trying to ask. And so I think it's really where you get those colliding molecules, and whether that's through seminars or one-on-one meetings or symposia, and hopefully we'll be able to return to in-person meetings sometime in the not too distant future.
Given my background in cancer biology, I was really interested in looking at the functional implications of microRNAs. So I think that would be, for me, a great collaboration.
We really try to focus on the unanswered and most important clinical dilemmas we face in taking care of patients.
It's all based on a small microRNA, right? So I always say these guys are small but mighty.